Ash dieback disease is a fungal disease that has been infecting ash trees. It's been sweeping across Europe over the last several years. It was first identified in the UK about 3 years ago. In the east of England, you're already seeing a lot of infection and ash trees being killed by the disease, and this is spreading across the UK now. Because there are a few trees that are able to survive the disease in Europe, we've decided that we would like to take the latest genomics and statistical genetics techniques to see if we could identify genes that we can use to predict the tolerant status of trees. By identifying individuals that are tolerant to the disease, we should be able to identify trees from across the UK to use to repopulate the countryside.
We started out with a diversity panel of around 200 ash trees, and these were provided by another member of our consortium who'd been studying these trees for quite some time, so they knew exactly how susceptible they were to ash dieback disease. We then sequenced the RNA from those ash trees, and we used a range of bioinformatic tools to look for genetic markers. And these genetic markers could be in the form of gene sequence changes or difference in the activity of each of those genes. With these 3 markers, we now have a test for the first time that allows us to go out into the wild and identify trees that should be quite tolerant to ash dieback disease. We can then use those trees in future breeding programs to try and repopulate ash in the UK. This technology has allowed us to produce a test to look for susceptibility to ash dieback disease, but we could equally apply it to other crop species and other plant species, perhaps with more economic importance, such as wheat or oilseed rape.